Hi.
Hi, Jeff. Good morning.
Morning, Professor.
How are you doing?
Fine, yourself?
All right. Another.
Good. Yes.
Wow, we have 44 participants. I think that means we are pretty much complete. I don't know if Fiona's there too. Hi, Fiona. Maybe we just get started then. Yeah, so good morning, everyone. Today in this blog course, we have a module on so you heard a lot about differentiation, you heard about cell types, you worked with anatomical methods and resources on how to look at cell types in the mouse brain. And one thing that we had already discussed in some of the previous lectures are these powerful transcriptomics that can be used to classify cells and distinguish them and also understand how they might be functioning. And what we'll do today then is to look into really more deeply into what kind of methodologies are there and what kind of resources are there to do these analyses. And we'll start with a lecture from Jeffrey Fusil. He's in PsyCore from the University of Basel and also doing a number of courses for students, for PhD and postdocs as well, for how to apply computational methods for data analysis. And then in the afternoon, or part of the morning, and then the afternoon, you'll be doing some exercises with data sets that come out of these technologies that Jeff will describe to you now. And the goal is actually in the end that you'll be comparing different data sets from a transcriptomic experiment, and then to identify a gene that you think could be really interesting to study, and then you basically write a little essay or proposal around how one could study it, why you selected it, and so on. But now we'll start with Jeff's lecture, and I just hand over to him.
Great, thank you, Professor. So I'm very happy to be here with you. I'm also happy to be interrupted at any moment. And I think actually Elisabetta is going to give me a hand moderating that. So at any point, if you have a question, in the chat function, you can, or in the participant function, you can do the raised hand thing on Zoom, and then Elisabetta can stop me. So please don't feel inhibited at any time. Also, if you're having a hard time understanding my volume, or if there's something unclear, then please just speak up. But so far, so good? Good. And can we also see the screen share? You can see the slides? Excellent. So indeed, I'm happy to be here to talk to you about this technology. It's actually a super exciting time to be participating in molecular biology in general, in large part due to technologies like RNA sequencing. And I should say that every time I look at one of these data sets, I find something no one's really ever seen before. Um, it's really the Wild West and an unexplored territory. So we'll start with a bit of a background to frame what it is we're actually talking about, the types of questions that we're trying to address, and then we'll slowly build up the pieces of the technology that are required to address those questions. So I'm assuming you're familiar with this dogmatic perspective on information flow in biological systems, let's say. We have the DNA, which is where the information is largely stored. So the process of transcription, we get RNA molecules, which are more mobile. They can be sent to different parts of the cell or localized to certain regions. And from there, typically they're translated into proteins, which affect metabolism, structural aspects of the cell. What I should say, though, is that that's a relatively narrow perspective on what's really happening. Biological systems should be viewed as networks of interactions, both with the underlying components of the organism itself, but also with inputs both from the environment and signals that come out of the developmental and physiological processes themselves. So the types of development physiology that this network of molecules is generating also has feedback on the system. So it's a network perspective that you should have about it. I should also say that the information flow isn't necessarily just linear from DNA to RNA to protein. We know that proteins can regulate themselves. Proteins can be regulated by the small molecules that they produce or acquire from the environment. Proteins certainly regulate RNA. RNA can regulate itself. We can have antisense binding of RNA, for example. And most of these molecules can also modify DNA directly as well. So it's a relatively complex network we have to consider. So when we're talking about RNA-seq technology, and we're looking at, for example, what types and what are the abundances of RNA in a particular sample at a given time point, we have to remember that we're looking at only one component of this network. Adding complexity on top of this, especially in eukaryotic systems, is this issue of alternative splicing, which we're not really going to get into, but I think it's important to add, especially since we're present here with the Scheifele group. Now, as I'm sure you're aware, in eukaryotic systems, the information for example, protein coding genes is somewhat fragmented within what one might call a gene. So here on the DNA strand, we have the schematic of a gene where we have exons interspersed with introns. And there's a splicing process after the RNA is transcribed from the DNA, whereby these different exons can be stitched together in different arrangements. And in this way, we can actually generate quite a lot of diversity from one underlying gene. And this is how, in something like humans, we get 20,000 some odd genes. But that is actually sufficient to lead to the really enormous cellular diversity that we have. This is part of the story, is that because of this alternative transcription or alternative splicing, we can end up with many, many transcripts. In fact, we don't even really have a great handle on how many transcripts are in any given human cell, let's say. And then from there, of course, there's different processes that can lead to various proteins, including the alternative splicing. Actually, even in eukaryotic organisms, we can get something called transplicing, where two separate mRNAs can actually get stitched together. So there's a lot of really crazy things happening that we don't fully appreciate yet. And this is what's underlying cellular diversity. So far, so good? Good. So with RNA-seq, which we'll get into, this issue of alternative splicing is something that can be addressed with this technology, but it's not something we'll speak about specifically today. It's something we can address during the questions, and certainly your tutors are competent to guide you more in this direction. But the types of questions we'll be talking about accessible by RNA-seq technologies is what is often called differential gene expression. And by this, we mean which genes are expressed at higher or lower levels in, for example, a treated versus control sample. So maybe we're interested in what's happening in the transcriptional machinery of this really obese mouse compared to this lean mouse. So this is the framework of questioning that we're going to be addressing today as I describe RNA-seq technology. To start with a basic overview of the protocol, what it looks like, and then we'll get into several of these steps in more detail. Generally speaking, you start with an isolated cell or tissue population. Now, currently we can actually sequence individual cells. This is so-called single-cell sequencing. It's a bit more complicated on the technological and statistical level. We won't get into the details about that. It's not so different from what's otherwise called bulk sequencing, where you have a population of cells or a tissue. So let's say the question then is, we have this set of cells, maybe it's a tumor sample, maybe it's a part of a brain or some other tissue of interest, and we want to assess which transcripts are present, which RNA transcripts are present, and what's the relative abundance of these transcripts within this population. And maybe the question then is, we treat this tissue with a drug, and we want to see what the effect of the drug is on this population of cells. So if we're interested in assessing RNA, we first have to get the RNA out, and we then, if starting from this population of cells, we crack them open and get the RNA out, we see that there's really a broad diversity of types of RNA that are present in the cell. So of course, there's a lot of ribosomal RNA. This is actually the bulk of the population of RNAs, is this ribosomal RNA. We have the typical or canonical protein encoding messenger RNAs, which are polyadenylated. And we have a whole bunch of exotic and relatively poorly understood RNA molecules, for example, small non-coding RNAs. We have long non-coding RNAs. There's also microRNAs. There's lots of transcription happening in the genome, which I said we're just beginning to appreciate now. So these populations have different physical properties at least. And there's not really a one-size-fits-all extraction protocol to capture all of this diversity of RNA molecules. So typically, you go after one set. So if you're interested in, for example, protein coding transcripts, you might be interested just primarily in the messenger RNA, in which case you can use a poly A selection protocol. This is actually affected by a poly DT, sort of complementary binding strategy. So these poly A tails will stick to your column, for example. If you're interested in the smaller RNA molecules, you can, for example, do a size selection strategy to get at those. If you're interested in more of a mix, what one can do is actually deplete the ribosomal RNA. We're generally not interested in ribosomal RNA, especially in eukaryotic organisms. I mean, if you're doing microbiome work or something like this, you might be interested in them. But generally, because this is really the bulk of the RNA in the cell, we generally need to get rid of it. Otherwise, all we end up sequencing is the ribosomal RNA. So if you do the poly A selection, that will get rid of the ribosomal RNA because it doesn't have the poly A tail. You can also do a ribodepletion strategy, which is where you basically bind up the RNA and let the rest of the other molecules flow through, in which case you get a mix of, for example, long non-coding RNAs and polyadenylated. There's quite a lot of different strategies here, but something you need to think about when you're starting an RNA-seq experiment. What are the target RNAs that I'm going after? So now you have your population of interest, and now we would like to actually sequence these to figure out what they are, and then ideally how many of them there are. The technology that allows us to do the sequencing requires actually that we're sequencing DNA. RNA itself is difficult to sequence. It's also quite unstable. So generally what we do is convert this RNA into complementary DNA. So then we have the cDNA. And then to sequence the cDNA, we have to stick some adapter sequences on the edges of the cDNA. This allows the sequencing just to proceed smoothly and allows us to keep track of what we're actually sequencing. So there's another step actually of PCR amplification that goes on here. And then basically we take this sort of pre-prepared cDNA, this labeled cDNA, and we stick it in a sequencer, which we'll talk about. And then the sequencer outputs a whole bunch of DNA sequences, which we call reads. From there, we need to then make sense of, where these reads came from. So we have to now compare back against a reference. So for example, the reference genome from which this RNA originally came from. So if this was a bunch of mouse cells that we started from, in the end, what we would like to do is take these reads that come out of the sequencer, compare them back against the reference mouse genome to figure out where all these reads came from. And then from there, we can do some downstream statistical analysis to make sense of things like abundance and maybe alternative splicing. Another cartoonish way to think about this. So here we have our cDNA, let's say, that we've started with, right? So we have a bunch of different cDNA that are here color-coded. We actually have to fragment these into smaller pieces because the cDNA can actually be quite large. We have things that are kilobases, megabases in size in eukaryotes. Those are very difficult to sequence, so generally we fragment them into smaller fragments. And it's actually just the ends of these fragments that usually end up getting sequenced. So let's say these fragments are 1 or 2 kilobases. It might be just the first couple hundred base pairs from the edges of the fragments that are generally sequenced. So we get a whole bunch of these shorter sequencing reads, and we generally have millions of these for an RNA-seq experiment. For a eukaryote, for a general idea about which genes are expressed and at which level, we usually want something like 30 to 40 million reads per sample. So it's quite a lot of data that we start with here in terms of these reads. And then we map these back against a reference genome. So I've shown here schematically, let's say this is chromosome 1 of the mouse genome, and we have a gene here that's in blue, and then next to it is a gene here in red, and we have this exon intron structure. The way the reads are going to map in this case, because they come from cDNA, they'll actually jump over these intron boundaries, which I've shown here with this thinner line connecting one of these reads. So the reads that came from within an exon, they'll just sit on top of the exon when you do the comparison, the so-called mapping. But those that span the exon-exon junction, you'll find that the read only matches until the end of this exon here, and then the read continues to match at the rest of the exon. So we actually need an algorithm that's capable of making sense of this in an efficient way. So far, so good? Excellent. What I've shown here is the abundance of sequencing reads as mapped against the mouse genome for one sample. This actually comes from a real data set that was published by Elisabetta. You can actually explore this data interactively on your own if you jump to this website. And what this is showing is all of the chromosomes for mouse. So of course, they're ordered from the largest to the smallest. And what we see here is this kind of histogram is the abundance of reads as they're spread across the chromosomes. And it might be surprising to you that it's not very even. So what this is suggesting is that there's actually hotspots of transcription along the chromosomes, right? So what this histogram is saying is that there's some number of reads that map to this region and very few that map to here. What this probably is the centromere, where there's not so much transcription going on, for example. But this is what one data set of reads would look like spread across the, let's say, the whole genome. If you were to zoom in, you would then start to see this exon intron structure that I showed you previously, like so, right? So we'll first talk about the sequencing technology itself. There are many strategies for sequencing. There's sort of 3 main families of these, one of which is sequencing by synthesis. And you're likely familiar with this from this Sanger sequencing, which is used generally to find the sequence of, let's say, a PCR product. The Illumina technology, which we'll be talking about, uses a related approach. This solid approach is no longer existing. There's some dead ends in sort of all evolutionary trees. And there's some new generation things like nanopore sequencing. I'll leave this to you to investigate on your own if you're interested, but this nanopore sequencing and this zero-mode waveguide from Pacific Biosciences is super interesting And in fact, when you read about the technology underneath this, it should actually blow your mind that it's actually possible, that we're doing sequencing of, basically single molecules within a protein complex, and we're sensing its sort of energy field. It's really wild what's possible now. But we're going to focus on this Illumina technology, which is also very interesting, but also happens to be quite cheap and very high throughput. So the sequencing with an Illumina sequencer starts with our DNA, or cDNA in the case of RNA-seq. And we stick these adapters on the end of these molecules. And what the adapter allows you to do is fix it onto the solid surface of the sequencing flow cell. So now we have our cDNA molecules fixed in place. And what we do is the so-called bridge amplification, where the second adapter actually flips over, also attaches to the surface, and now we can amplify across in the other direction. So here we have a single strand, which then folds over, and we can then generate the second strand. And this happens a whole bunch of times. And what we end up getting is a cluster of copies of a single piece of cDNA in one region on the flow cell. So we started with one piece of cDNA that came from an mRNA. We stick a single copy of it onto the surface, and then we actually amplify it so that we get a better signal around the area, because we're actually going to visualize this directly. You're literally going to take a picture of it to see it. So to be able to see it well enough, we actually make many copies of it in one region on the flow cell. But this cluster here all comes from one original cDNA. So to actually figure out what the composition of this cDNA fragment is, we use this sequencing by synthesis. And the idea here is that we add one nucleotide at a time, right? So we have this strand, a single strand, and then as we're building the second one, we add in a base, we wash out all the unbound bases, and then we detect the signal that's attached to this base. So they're all fluorophore labeled. So each of them has a different color, the 4 different bases. So we add a T, or we add the next base, we wash it off, take a picture, and then we add the next base, take a picture, and so on and so forth. And so we basically have a picture for each one of these as they elongate. Does that make sense? So we're really doing base by base sequencing here, and we use the Terminator sequences here to get at that. So really what we see then is this picture, right? So this is one of the snapshots. So this is the surface, and this is the cluster of one of your cDNA molecules, for example, with this little green dot here. So this all came from one cDNA that got amplified. And so the raw data for this Illumina style is actually a whole bunch of these pictures. So it's really terabytes of data per sample that we start from. The instrument then is able to keep track of what's happening in each of these clusters. So you get this kind of movie, and these are the different frames of the movie. And then these frames then correspond to the DNA sequence, for example. Is that clear to everyone? I don't think I see any questions so far. So the images actually get deleted after we generate this because it's really way too much data to keep track of. So in the end, what we're really just keeping is the DNA sequence and then some quality information about the sequencing itself. So a brief recap here. We've started from our RNA population of interest. We converted it to cDNA, and then we sequenced that. And that sequencing process also has some PCR amplification steps that are needed for the sequencing process itself. So what we end up with is DNA sequences, and it's at this point already fairly abstract from the underlying RNA population. So in RNA-seq, we can't really directly make a comparison between what we sequenced and what was in the RNA population because there's PCR amplification steps in between. And there's some biases that go along with that. So one of the misconceptions about RNA-seq is that we're getting a direct measure of the number of RNA molecules per cell. And that's not the case. We have to take a more relative abundance approach. Just something quite important to keep in mind. But the questions then are, we have all of these reads. Now, how do we make sense of where they came from? And then how do we get at this question of the abundance of the sequencing reads? So that's what we'll get into next. So for this first question, we can use some other little tricks with the sequencing itself. So we can actually keep track of the strand of the gene from which these RNA molecules came from. So to get at the problem that this is actually addressing, just have a look here at this schematic of, let's say, 2 genes on the chromosome. So we have a gene here in green, and this gene is actually transcribed from left to right. Yeah, so it's going in, let's say, a 5' 3' direction. Well, they always do, but on a chromosome, you have 2 strands that go opposite directions, and you can actually get transcription from either strand. Right? The genes can have different orientations. This is what we would call a plus strand orientation, and then this gene here in brown is a negative strand orientation. Is that clear to everyone so far? Good. So to make things even more complicated, because that's what it's like with eukaryotic organisms, there's actually a third feature here, shown in yellow. Maybe this is one of these long non-coding RNAs, for example. Now, if we have a library where we don't keep track of which strand the RNA molecules came from, we can't really make sense of this problem here, for example. So in this brown gene, it's transcribed actually in the negative sense, so it's going from right to left, and this yellow gene is going left to right. So when we have our sequencing reads and we, let's say, map them back to the reference genome, this is where they land. There are some reads here that overlap these 2 different features. And from this perspective, we can't tell whether this particular read here came from this yellow guy or this brown guy, for example. It's equally likely that it came from either. However, if we can keep track of the strand from which the RNA came, because remember, RNA is single-stranded, we can now sort of deconvolute or deconstruct where these reads came from. So the color coding for these reads here in blue and red, this is the different strands that they came from here. So if we have a stranded library where we can actually tag this from the beginning, what you could see in the end is something like this, right? So all of the reads that map to this particular region on the genome are in this blue sense, the forward strand sense. And that's consistent with what we expect for this particular gene because it's transcribed left to right. And then here, where we have this overlap, we can now figure out that, okay, all these blue reads actually belong to this yellow feature because it's going left to right. And all the ones that are here in red actually belong to the brown feature, which is going right to left. This is achieved by a trick with the way that the adapters are ligated before cDNA synthesis. We can actually tag the cDNA molecules to figure out which strand they came from. So this is an important trick for RNA-seq. Something else that you can do is so-called paired sequencing or mate pair sequencing. What this allows you to do in both cases is to assess a larger fragment of cDNA. So they come in all different sizes, these fragments. And again, let's say these fragments are, I don't know, 800 base pairs or a kilobase. You're generally only sequencing a couple hundred base pairs, right? So Illumina sequencers, you're generally looking at 150 to 300 base pairs. So you can only really get the edges of these fragments in any case. You can use a so-called paired end approach where you can actually sequence from both ends of these fragments. So you actually get 2 reads per fragment. This is so-called paired end sequencing. And since you know where these 2 reads came from, you can actually infer, how far apart they are from each other, and this helps you place them onto the chromosomes. Mate pair sequencing is similar, except you actually make a circular molecule, and then you fragment and you sequence from the edges out this way. So you still get information about a larger molecule, but it's just a different library preparation.
Maybe, Jeff?
Yep.
Maybe just to all the students, I think now the next part, this is all the concept of the method. Right now, the next part, Jeff will go into sort of more from the computational workflow on how this is being analyzed. So this would be a good time if if anything's unclear about how this actually works, how the procedure works in principle, just maybe type a question in the chat. Or if you have all perfectly understood everything, you can also type that into the chat. And because now I think Jeff is going to change gears a little bit, and it's going to be about file formats and so on. So if you have questions on this first part, now is the best moment to raise them. Or you give us a thumbs up if you got everything perfectly. I always ask that in the Grundvorlesung. If I hear silence, does that mean yes or no?
There's a couple thumbs. I mean, it is pretty complicated. So certainly if you have questions about it, you're not the only one. And it is a good time to talk about it. Yeah. We can also come back to it. Otherwise, I guess we forge ahead.
Okay.
All right. Okay. So we have our raw data, which is a whole bunch of DNA sequences. The workflow looks basically like we do some first quality assessment to make sure that the data was generated in a sufficiently accurate way. We may need to do some filtering or trimming of the data based on this quality control. So we have our filtered raw data now, and there's sort of two approaches one can take with RNA-seq data. Now, generally, if you are working in a model organism and you have a good reference to compare against, you can now align or map the reads back to this reference genome. And from here, you can start counting the number of reads per gene, doing this differential expression, trying to figure out, what types of pathways are affected, and so on and so forth. You can also use these reads and generate a de novo assembly, which means you can actually reconstruct the transcriptome from the reads themselves, which you can then map the data back against. So if you're working in a non-model organism or something novel, this is usually one of the steps that you have to go through. But basically, we take our raw data, do some quality control, map against the reference genome or transcriptome, and then start trying to make sense of which transcripts are there and at what relative abundance. There are several file formats involved. The sort of two main ones are this FASTQ file and this BAM file. Now, the FASTQ file is what you get from the sequencing facility, basically from the sequencer. So hopefully you're familiar with the FASTA or the FASTA format, excuse me, which is generally used to store DNA or nucleic acid information, where you have one line that's a header, and then you have the next line that is the actual sequence itself. FASTQ is very similar to that, except it has an extra line of data, which is the quality score for each base pair of the DNA sequence. That's what the so-called FASTQ file is. So you can use that quality score for each of the bases to do this quality assessment and do some filtering and trimming if you need. And then once we have these reads that we're happy with, then we align it against the reference genome. And we end up with the so-called BAM file. And this is a binary format that keeps track of, for each read, where they map to within the reference genome itself. So it has all of the information in the FASTQ file, plus the chromosomal coordinates of where it maps to on the reference genome, if that makes sense. If you're interested in looking at variants, you would generate something called a VCF format, but we're not going to talk about that today. So here's the FASTQ file. This is the header. This tells you information about the sequencer that it was produced on, some other standard technical information about the sequencing itself. Then you have the actual sequencing read. You have this third line, which is generally useless. It's like a separator. And then this fourth line here is now the sequencing quality score for each one of these base pairs. So you can see that like this C, for example, maps to this F here. Here it was ambiguous, and so you get an N because it wasn't clear what the base pair call was for this case. And here it's this this hash symbol. So this is this ASCII encoded quality score, so-called Fred score. It's a probabilistic based, and what it's basically telling you is the probability that this base pair is correctly called. And it's encoded with this alphanumeric. With paired data, you now get 2 separate files for forward and reverse. So you get this R1 and R2 files per sample. This is how you generate the probabilities from it. It's not so important. If you were to look then at the distribution of these quality scores, so let's say we have 100 base pair reads in a particular sample. You can look, for example, at the distribution of the quality of the base pair calls to each of the individual base pairs. So this is a summary of, these are box plots. So this is showing, let's say, the distribution of quality scores at position 1 across all of your millions of reads. So on this sample on the left, what it's showing is that, for the first 60 base pairs or so, you have quite good quality scores. So they're all, sort of 30 and above, which corresponds to high quality. And then towards the end of the sequencing reads, there were some problems. So there was maybe something that happened with one of the amplification steps, or something problematic with the reagents or with the flow cell. And you can see that you have very low quality at the edges of the reads here. So in some applications, you might want to actually trim this data off of the edge of these reads because they're lower quality here. And this sample on the right, we see that we have consistently high quality scores across the whole length of the reads. So this is the type of quality control that one should do. No questions there, I guess. So we have our high quality reads, and now we want to map these against the reference genome. So when we're using whole genome resequencing, there's a particular set of algorithms one can use because the reads pile up kind of right across the chromosomal sequence because we're looking at all of the DNA. When we're talking about RNA-seq, we've started from RNA molecules converted to cDNA, and now we're taking reads from the cDNA and aligning these back against the reference. So we don't get this even distribution, but we get reads that pile up on exons effectively. And we need different algorithms to do this in an efficient way. So STAR is pretty popular and quite accurate and modern approach to do this. The reason why we don't use something simple like BLAST is because basically it's really way too slow. BLAST is the algorithm that you use generally for finding similar sequences. So if you have, sequence of interest, you want to know what other organisms it comes from, you can use BLAST, but it's not fast enough actually for the problem at hand. You don't need to belabor that point too much, but it takes, actually for STAR, you can do one sample within something like 20 to 30 minutes, depending on how many reads you have. But these other algorithms take hours and days to do that. It's not super important to understand the algorithm itself, but basically you make an index of all of the exon junctions in your reference. And this allows you to very efficiently look at, kind of prefixes of the reads. So kind of fragments of the reads and where they might fit on an exon, and then stitch them together in an effective way. And I leave you to look at the literature for this to understand the algorithm better if you're interested. So an important aspect of RNA-seq is considering the genome that you want to use as a reference, right? So we have to compare the reads that we have back against something to make sense of where they came from. So you have to choose this wisely. Some of the things you have to consider are the annotations that come with the reference. So is your reference genome accompanied with a good set of annotations that say where all of the genes are? Are the exon-intron boundaries well defined in this particular reference genome that you're using? If you're interested in humans, for example, the idea that there's one reference genome that's representative of all of humanity is fairly absurd, right? There's separate, evolutionary processes for all, populations of organisms. And so if you're interested in looking at, for example, this is more relevant, let's say, for looking at mutations, but if you're interested in, a really niche population like, some strange pygmy mouse from a far-flung island, it might not make sense to use a standard lab strain of the mouse reference genome. These are often encoded what are called alternative scaffolds, right? So take-home message here is that the reference genome that you're using should match the organism from which you actually did the sequencing. Let's put it that way. And there's this step of creating an index so you can do the mapping efficiently. And this has to be done for each algorithm separately. So the index that you create for star isn't compatible with, a different algorithm. And you have to use the annotation file that accompanies the sequencing data itself to generate these indices. So you need the FASTA file of all the chromosomes, and you also need an annotation file saying where are all the genes on this chromosome and where are the exon-intron boundaries. Any questions about the reference genome? It's a pretty complicated topic, but that's just the essence of it, yeah? Okay. So now we have all of the reads mapped against a reference genome or reference transcriptome. And now we're interested in quantifying these reads. So we want to get at now, is gene A expressed higher than gene B? And we can use the abundance of the reads as a proxy for getting at that information, right? So we now want to start quantifying the number of reads as they correspond to different features on the genome. And by features, we generally mean genes or transcripts, yeah? It's important to remember that underlying this are stochastic models, right? So we're assuming that we're sampling stochastically from a population of RNA molecules. And so more abundant RNA molecules will be sampled more frequently than low abundant RNA molecules. And larger RNA molecules will be sampled more frequently than smaller RNA molecules. Now, we go through this process of aligning them against the reference genome or transcriptome. Now we can start to count the number of reads that align to these features, right? So we're assigning these fragments to the genome. One of the complications we run into here is actually that any particular read might align with equal probability to different regions in the genome, right? So we have, for example, multi-copy genes where the sequence is really identical. These may even be present on different chromosomes, for example. We also have genes that recycle some information, right? So we think about domains being conserved in different proteins. It might be that gene X has the same subset of sequences that gene Y has. And since we're only looking at short sequences here of a couple hundred base pairs maximum, it might be that in some cases we can't distinguish whether any particular read comes from gene X or comes from gene Y. You need to figure out what you want to do with these so-called multi-mapping reads. So once we have these read counts per gene, we need to convert these to some sort of relative abundance somehow. And what we need to account for are differences in the library size. And by library size, I mean in sample 1. And of course, you should have replicates for your, so let's consider the simple case of our fat and our not so fat mouse. We need minimally 3 replicates from the fat mouse and 3 replicates from the not so fat mouse, yeah? And so we have, let's say, minimally 6 samples for this simple RNA-seq workflow. Each of those samples is going to have a slightly different number of reads that were sequenced. So maybe in sample 1, we have 47 million reads. In sample 2, we have 45 million reads, so on and so forth. And so we need to account for that so-called library size, yeah? Otherwise, we can make erroneous conclusions about how abundant things are. We also have to consider the lengths of the genes we're talking about, because as I said, we're more likely to sample from larger transcripts, right? So if we have a really long, let's say megabase long RNA molecule, we can actually get a lot of fragments of DNA from it because it's so large. And you may erroneously overestimate how abundant that is compared to a very short molecule from which we can only get a smaller number of fragments. So we need to somehow normalize for the length of the transcripts that we're talking about as well. And we should make some attempt at sequence-based biases which are factored into the amplification steps. These are some of the technical aspects that need to be considered. Any questions there? There's a lot of actually deep technical knowledge that's required to do these algorithms correctly, but we certainly don't have time to get into that today. These are just sort of the topics that one needs to be aware of. Good news is that it's largely a solved problem. So another thing to consider is that, RNA-seq comprises many technologies which are rapidly evolving. And so if we're talking about the Illumina style sequencing, that needs to be treated differently than if we're talking about long read sequences from, Pacific Bio sequencers, for example. Depending on what library prep you used at the beginning, you have to consider how that may affect how you're doing your read counting. And you need to be, aware of the questions that you're asking. And this also will affect, which particular protocol you use. What's really crucial is that your gene and transcript model annotations are accurate, right? So if you have a poorly annotated reference, you're going to not be able to make sense of what it is you're looking at. So we need to figure out when we're doing our read counting, what to do with the multi-mapping reads. The simple solution is just to discard them. If we can't accurately decide whether this read maps to gene X or gene Y, a safe thing to do is just get rid of it. What this looks like actually, just schematically, so imagine we have this case of overlapping gene 1 and 2. We can't disambiguate where these reads shown in gray belong to. So one solution is just to ignore them and only count based on the unambiguously assigned genes. Yeah. In the case of using transcript models, you may be able to disambiguate some reads. For example, we have 2 transcripts coming from a single gene. Maybe this transcript A only expresses exon 2. And so we know that any reads that map to exon 2 are specific to transcript A, right? They can't belong to transcript B. And equally, in this case, if we have reads that span the exon 1 to exon 3 junction, we now know that these belong specifically to transcript B. So these are some tricks that you can use to disambiguate a bit. So indeed, the simplest approach is actually just to discard the multi-mapping reads. You lose some information, but not a huge amount in most cases. You can introduce some biases, but this is actually an appropriate approach for the standard workflows. And it's actually not that much of a loss of information, and it's the safer way to go about it. And at least it's clear what it is you're talking about. You can try to rescue these multi-mapping reads by the so-called fractional allocation. And the idea behind this is to estimate the abundances based on the uniquely mapping reads, and then divide the ones that are ambiguous, based on this distribution of the uniquely mapped reads. It's complicated to do it, but basically we could, make some distribution of abundance of these uniquely mapping reads. So maybe we have like a 3-to-1 ratio between transcript A and transcript B. So then we could allocate these ambiguous reads in a 3-to-1 ratio, for example. Some people try to do this, but it's really problematic because there's lots of exceptions and it's not clear what the algorithm's doing in many cases. So you can get a little more information out of your data, but I think you introduce a lot of errors by trying to do this. So generally, we just drop the multi-mapping reads. Now, there's another question.
Sorry.
Yeah.
I thought we're 45 minutes in, and it's a lot of information to digest. Maybe this is the moment for everyone to, including me, refill your coffee cups and follow whatever other bodily need you may have. And then we just continue in a few minutes, so maybe at 9:55. Would that be good for everyone? I see one thumb. I see a nodding face. Okay, excellent. No, great. So then I think everyone has a chance to digest a little bit. And let's see you all back at 9:55. Okay. I hope everyone is refreshed. I see a few fresh faces. Hello, everyone, back again. I think let's keep going. Okay, Jeff, you're
Thank you.
there. Over to you again.
Thank you. Any questions occurred during your little break? Just throw your hand up and Elisabetta will stop me. So to maybe bring us back to where we left off. So we've got our RNA, we convert it to cDNA, we do the sequencing, we got a bunch of sequencing reads, map them back to the genome or reference transcriptome, and we have some of these problems we need to sort out, right? It's more complicated to look at the transcript level than looking at the gene level. But if you're looking at the gene level, what you would have to do is, consider one, let's say, super transcript for a particular gene. Now, there's some trade-offs for each way of looking about this. So let's say the real situation looks something like this. So for one gene, we have two transcripts, transcript A and transcript B. In this scenario, if we're comparing, let's say, condition A to condition B, so this is control and treatment, for example, let's say we see that both of these transcripts decrease in abundance if we have transcript level resolution. If we're looking only at the gene level, so let's say we're just considering one super transcript for both of these transcript models, in this case, we still see the same dynamics, right? Because actually both of the transcripts are going in the same direction down. From condition A to B. If we're looking at the gene level, we would still see the same dynamics. Now, there may be situations where we have a specific transcript of a gene that behaves differently than other transcripts. And this certainly happens, especially in eukaryotic organisms. So imagine the scenario here where transcript B actually increases by a couple fold. Transcript A more or less stays the same. If we were to combine all of these reads from the two transcripts into one super transcript or to one gene level model, the dynamics we'd actually see here would suggest that there's actually not much difference in this case, right? Because these have sort of opposing effects or different effects that get sort of muted out. Does that make sense? So you get,
Sorry, Jeff, maybe just to make that extra clear, right? So for the students, you heard all from the introduction from Jeff, right? That one gene, of course, can produce many different transcripts, right? Through using different alternative promoters or differential splicing and so on. Mm-hmm. And the important thing here is that even if you may not want to care about the individual transcripts, you could get completely misled about interpretation of what happens with the gene as a whole if you don't separate in your model what you actually quantify. Right? I think that's the really important point here. So one gene, many different transcripts, and they can have completely different dynamics. And these short reads that you get with the RNA-seq are not necessarily easy to allocate or to know where they're coming from, because they could come from a common part or they could come from a unique part. And that's sort of the big problem here.
Exactly. Yeah. Thank you for that. So it's much more complicated to get at the transcript level resolution with short reads because, in many cases, these different transcripts share a lot of exons. So then you have a lot of multi-mapping reads you need to sort out somehow. But, you will capture this type of dynamic if you're able to do that. So it's indeed more informative to look at the transcript level. It's very complicated. So one of the key guys behind a lot of these methods, Lior Pachter, University of California, it's a funny link and actually talks about how the methods behind this framework, cufflinks to do this transcript level counting was actually not even really reviewed properly. It's super complicated statistically. It generally introduces a lot of extra noise. The good news here is actually that there are new types of sequencers coming out, for example, from Pacific Biosciences, that allow us to sequence kilobases of reads, right? So we can actually sequence the entire transcript now. And then we don't have to worry about this issue with the short read sequencers like Illumina, where we have a lot of multi-mapping reads. It's still too expensive to do this routinely, but the prices are dropping fairly rapidly. So when we count at the gene level, we're collapsing the reads from all.
I have a question, sorry.
Yes, please.
To identify if a read is uniquely to one transcript, are there reference transcripts, or how is that done?
Yeah, so okay. It's a bit different depending on which organism you're talking about, because there's different research communities behind each of these different organisms. So for example, in mouse, there's a group of researchers who maintain the references for the mouse genome. And when we talk about a genome reference, there's always these annotations that come along with it. And the annotation files then specify, where all the exons for a particular gene are. So basically the information for all these transcripts are encoded in these annotation files.
Okay, thanks.
Yeah. Right, so gene level counting is preferred because it's more interpretable, and performance-wise it's much easier to do. So it's simpler just to drop multi-mapping reads and look at the gene level. You can have some problems with type 1, type 2 errors when we do gene level counting, but based on real and simulated data sets, it's relatively minor effect. And if you want to be super accurate, this paper here talks about how to incorporate some offsets from transcript level estimates. This is a fairly advanced application, but if you're interested in sort of the cutting edge of where RNA-seq is at, this is a good place to start. So let's get back to the question that we framed at the beginning then, which is, let's say we're interested in which genes are expressed at higher or lower levels in treated versus control sample. So now let's say for a particular gene, X, let's say we have 3 replicates for condition A and 3 replicates for condition B. And this is how many reads map to that particular gene. Now we have to figure out, between this set of replicates and this set of replicates, is there a statistically significant difference between these two? So in this particular case here, it's pretty borderline, right? I mean, what the issue here is that there's actually quite a lot of variance between the replicates. So your confidence in saying that, in condition A, the gene X is higher than condition B, it's not so obvious in this case, right? And we need a proper statistical framework to go about this. And of course, we need to do some kind of normalization as part of that to deal with things like differences in library size. It might be that for this particular sample, you had 60 million reads, and for this particular sample, you had 40 million reads. And maybe this is just what you're seeing here in this difference in total reads. There's sort of 2 approaches to go about it. One which is non-parametric based on linear modeling, and then there's a parametric approach. And these are the common software packages which are implemented in the R statistical software, which you'll be using later today. You won't actually have time to look at the differential expression workflows themselves, but if you're interested, there's very nice vignettes to follow for these. The assumptions here are that actually most genes are not differentially expressed. That's an important part of the model underlying these. And the other important part of the model is that it's equally probable for a read to map to gene X in all of your samples. So there's nothing saying that in your obese mouse that you have biases in terms of which genes are present, or the structure of the genome basically has to be the same between your samples. That's what we're assuming here. So you couldn't, for example, really compare a mouse and a rat. It would be really complicated using these statistics. It wouldn't really make any sense. So we have to do this normalization based on library size, the length of the gene, biases in composition. The actual methods for doing this normalization are quite complex, and you really need to follow quite a lot of mathematics to grasp what exactly they're doing. You're welcome to do that, of course, in the in the primary literature. It would take some hours to really look at properly. They're different methods, but they use similar ideas. But what's encouraging is that, they're pretty comparable. So if we look at EdgeR and DESeq2, looking at type 1, type 2 errors, there's a little bit of a trade-off between specificity and sensitivity between the two. But generally, you're going to get the same answer from these different methods. So that's the good news. Now, when you have, let's say, your normalized reads, now you want to look at whether there's a statistically significant difference between your read counts for your replicates of condition A versus B. There's several different approaches to doing this statistics. What you can't really do is just a t-test, because actually your variance estimates for 3 replicates are very poor. So a lot of what these packages do is take advantage of the number of genes you're looking at to do some corrections for your variance estimates. But in the end, you do something like a t-test to look at the difference between condition A and condition B, and then you get a p-value from this test. And then, of course, you would want to do some kind of moderating of your p-values. So a p-value at 0.05, if we had, let's say, 20,000 genes that we detected, if we're saying that our threshold is 0.05, that means that in this scenario, we would expect 1,000 significant tests, even if there's no differential expression, right? This is the meaning of a p-value. So we need to do some kind of moderating of these p-values to deal with the number of tests that we're doing. If you do 20,000 tests and you're expecting that 5% of them will be erroneous, then you end up you're expecting quite a few erroneous tests. So then you have to take these p-values from these tests and then do some adjustment on them as well. It's fairly well understood statistics. It behaves pretty well. And then the good news, the take-home here is that the RNA-seq workflow, which includes this downstream statistical processing, is highly concordant with independent methods. So if we take, for example, QRT-PCR and compare that against what our abundance estimates are telling us from RNA-seq, in many cases, the validation rates are really high. So what this is telling us is that RNA-seq data, even though we're starting from RNA, going to cDNA, and we have to do all this normalization and some kind of obscure statistical tests, what we're getting in the end is reliable because we can use independent methods to validate what we're seeing. That's the good news. Okay, so we do this whole thing, and we now have a list of genes that are differentially expressed between our obese mouse and our not so obese mouse. Okay, so now we have a list of genes, and then what do we actually do with this list of genes? How do we make some sense of the biology from here? We all good so far? No questions from here? Okay. So one of the things we can look at is the annotation of these genes, right? So, what are the functions of these genes? What pathways do they belong to, right? One of the resources that's really handy for doing this is Biomart from Ensemble. And what you can actually do on this website is give it a list of gene identifiers, and then you can get all sorts of descriptions and annotations out of this with a few clicks, right? You don't need any programmatic skills to do this. So one of the things you can do from here is, look at the annotations of whatever genes are differentially expressed. That's a good first start. What you might also be interested in then are, you have some range of fold changes, right? So maybe you have some genes that are 10-fold difference in abundance, and maybe some genes that are 1.5-fold difference in abundance. You can try to do some cutoffs and only look at things that are really strongly differentially expressed, so things that are maybe 2-fold and higher. That's a common approach. What you have to keep in mind when you're doing this is that the abundance of a transcript doesn't always directly relate to the abundance of the encoded protein. Sometimes it's actually the opposite. They're inversely related. And that doesn't necessarily inform anything about the activity of the encoded protein product. So it's one approach that you can do to look for really large changes in transcription, but that might not actually relate directly to changes in biological activity in the cell, just to keep that in mind.
Jeff,
yep.
Just to very briefly intersect and
yes.
get everyone's attention on this. Basically, in the exercise today, this is sort of where we're jumping in, right? So all of the workflow that Jeff just described has sort of been done for you, and you get your list of differentially expressed genes and fold changes, how they're different between two conditions, and p-values. So all this discussion that comes now from Jeffrey is things that you should really keep in mind then when you're looking at this data in your exercises that you do with R.
Thank you. Okay, so fold change cutoffs can be useful, but take it with a grain of salt. It's often more complex underlying biology. What some people try to do is separate genes that are upregulated versus downregulated, right? So genes, for example, that are expressed at a higher level in the obese mouse, or genes that are expressed at a lower level in the obese mouse compared to the control. Something to keep in mind with this is that, in pathways, biological pathways, we have positive and negative regulators, right? So if we have an increase in a negative regulator, that might actually mean that the output of the pathway is decreasing. So let's say the transcript is higher in the mouse, but unless we relate that to what we think we know about the encoded function of that transcript, it's a bit difficult to separate things into up and down. It's not entirely useless, but it's sort of a complicated way to think about it. Another approach people use is whether there's a statistically significant enrichment of certain annotations. So let's say we find that there are 300 genes that are differentially expressed in our obese mouse. We could say, for example, are there certain pathways or certain types of annotations that are observed more than random in this particular mouse? So maybe there's something related to metabolism that's quite interesting in this particular mouse, or maybe something related to, hormonal imbalance or something like this. And there's some statistics behind doing this as well. The two approaches that you're going to be investigating later today involve, some visualization strategies for this at the sample level themselves. So can we start to make clusters of, where our sample groups relate to each other? So we're going to look at two different techniques. The first one is called principal component analysis, and then we'll look at something called hierarchical clustering. So principal component analysis has been around for a very long time. And the idea here is to reduce the number of dimensions in your data. And by dimensions, I mean that, let's say we have an RNA-seq data where we've reliably detected 16,000 genes, right? So we now have a matrix of 16,000 genes times whatever number of samples that you have, right? So it's a very high dimensional data set. And it's difficult to visualize that, right? I don't think anybody can think in a 16,000 dimensional plot, for example. So what we can do is actually reduce our data to a smaller number of variables. And we can do this mathematically. And so we take our large matrix and we project that onto a smaller number of dimensions, right? So we can take all of the variables of our data, which in this case is, the expression levels for each of our genes, for each of our samples. So you can think in a table format here, where we have in the columns all of our samples, and in the rows are all of our genes. And what we can do is make linear combinations of these variables and collapse them down to one dimension, right? So let's say X is all of our genes. So you have X1 all the way through up to P. And we can make some kind of, parameters for each one of them, so-called weights. And we can make a linear combination of these that are now captured in one variable, this so-called super variable. And we scale these so that they're equal to 1, which means that each of the genes, has some contribution to this particular variable. So a gene that, has a lot of explanatory power at this particular variable would have, a high weight here, so a high fraction, and others would have lower, but they all sum up to 1. It's a bit complicated to describe mathematically if you're not a mathematically oriented person. So I think what I'll actually do is just jump to the graphical representation of this. One important thing to keep in mind is that this procedure is unsupervised, right? So we're taking all of our samples and we're trying to do clustering without using any information about what we know about these groups, right? So we don't have to label the data and say that I know these are the treatments and these are the controls. We treat them all equally, yeah? So let's just skip to the graphical representation because I think it's an easier way to describe this. So imagine a simple case where we have two-dimensional data. In RNA-seq data, we have 16,000 dimensional data, but imagine we have two-dimensional data. And our two-dimensional data has some distribution like this. The way that PCA works is that we want to draw a new dimension in the data that maximizes the variance of our observations, right? So this is what the actual data set looks like. And we want to draw a new dimension that maximizes the variance. And as it turns out, if we draw a line this way through the data, we get actually quite a lot of variance from this line here, right, which is shown here in the blue. Also in this case, we can draw a line this way. And what the blue is representing here is sort of the distance from, this new dimension that we've generated and the data itself. So we take two-dimensional data and we project it onto this one single dimension here, right? So we collapse all of these points onto this red line. And so with principal component analysis, what we're doing is imagine taking this red line here and sort of rotating it around in your data until you find the orientation that maximizes this variance, right? That maximizes this spread. And then we can do this some number of times, right? Generally, we only do this, for the first 2, 3, or 4 times, because ideally you capture most of your variance in some number of these new dimensions. And another way that we call these dimensions, these are actually the principal components, let's say, or these so-called super variables, right? That's what we call principal component analysis. They're also called eigenvalues on eigenvectors, but that's a mathematical representation. So you can imagine taking your 16,000 dimensional data and whatever.
Can I ask a quick question?
Oh, sure. Yeah, no problem.
On this slide,
Yep.
Wouldn't the highest variance be actually like 90-grad opposite of the one in the lower left? So like if we actually did it like this time on the data, it sort of goes like vertically. Wouldn't higher variance be if it went sort of horizontally, like diagonally, but the other way around? I don't know how to explain it because like I can't point to the screen, but like now this line does not show the highest variance, does it?
Actually, if you want, we do a Zoom experiment. If you go to view options on the top of your screen, there is an annotate button.
And you can go to draw.
I'm using my phone right now, so I'm not sure.
Okay. Then
you can draw like this, for example.
Okay, then forget it. Sorry.
Yeah.
I think I know what you mean because I asked the same question. And what Sandro want to say is that you draw the line from the picture in the left from one lamp to the other.
Yeah.
That would be, then you would have the highest variation just by visualization.
Exactly. Thanks, Fabio.
I think, Jeffrey, I think they mean something like this.
Yeah. It depends on, how you actually draw the distances, right? So, sorry, my mouse is now frozen up. I don't know why. Uh-oh.
Maybe Julia,
Okay,
you have to report. Okay.
I'm back. Can you guys see my cursor moving? Is this okay?
Yes.
Yeah. So one way to think about it is, you're drawing a distance from here to here, for example, right? So the distance for this particular measurement to this hyperplane, to this principal component, is actually from here to here, right? Now, if we drew a line this way, the distances between the axis would actually be, maybe I can actually draw this. See if so we draw it from here, let's say. It's a very poorly drawn line, but our distances look like this. And then we sum them up, right? So these are actually pretty short distances here. Now, if I clear this.
So effectively, I think, Sandro and Fabio, what you were saying, in a way, you're completely right that this direction that Jeff is now drawing the line in, that's the direction where there's the biggest change. where there's the biggest variance. But the way it's in the PCA, it's basically you project it down, and the variance that's basically calculated in the PCA are these blue lines that are drawn. So it's exactly that direction that you had in mind. You see what I'm trying to say?
Yeah, exactly. You had the right idea. So you see the lines that I'm drawing here are what you intuitively
understand now.
Yeah,
Yeah.
it's just it's an algebraic issue of defining distance, right? And so you define this line here that lost my mouse again.
It's sort of how far is each open circle away from the blue circle. And by placing blue circles the way they are, you capture exactly that distance that you were referring to that you think is the biggest variance here.
Thanks for explaining. Thank you. Now I get it.
Excellent.
So I think that the confusion, can you hear me?
Yes.
It's because maybe a student thought, I don't know if, okay, you can see me, is that they were looking like this as a variance across the points. And so in this case, it would be like the minimal one. I think that was the confusion.
Yeah.
Just them from here.
So we use these distances here to, using my mouse again, sorry. Use these distances here to actually calculate what the captured variance is, and then we collapse all of these points onto this new dimension here, right? So we have two-dimensional data, these open circles, and now we have one variable, right? So we've taken all of our 2D, all of our 2D points, and now we have a one-dimensional vector here. This is what's meant by dimensionality reduction, right? Taking multi-dimensional data and now representing it as one dimension. So we can do this, X number of times until we capture, as much variance as we can in the data. And then we have some number of principal components. Now, if we want to look at just a 2D plot of these principal components, we can actually now just take, let's say, the first two most explanatory principal components and then make a plot based on these, right? So each of our samples now has a new coordinate system based on these principal components, right? And now we can compare these against each other, make new XY coordinates. And that's what, a principal component plot looks like. Just clear these. Hold on. You can actually calculate the amount of explained variance as part of the algorithm. So what this is saying is that the first principal component captures about 37% of the variance in our data set, and the second one captures about 18%. You can look at the distribution of these different principal components. This is a so-called scree plot. But what we now see emerging, each one of these points is a sample, right? So this is one from neocortex, from this kind of VIP type cell. And what ideally we see is that these are replicates and they cluster together, for example. And now we can start to see some interesting patterns, right? So, for example, all of the open shapes, these are all glutamatergic ones, right? And we see that they actually separate along this axis. So all the closed shapes are on the right side and all the open shapes are on the left side. We actually see there's a separation between GABAergic and glutamatergic, right? We can also see that these somatostatin positive cells cluster closer together, even though they come from different parts of the brain. Now, none of these labels here were used in the calculation of the principal component analysis. These are annotations that we can apply subsequently to make sense of it, to do some interpretation. So it's an unsupervised approach, this principal component analysis. So it's useful for quality control to be sure that actually our replicates cluster together. If we saw a big ball of all of our samples sitting together somewhere in the middle, then we might think something went wrong with our experiment, or we're actually just sampling from the same population. Well, what's underneath this is actually, all of the genes that were measured basically have a weight in each of these principal components, and they kind of push the samples along these different dimensions, right? And so each gene has some contribution to pushing these samples along each of these axes. So you could actually use the weights of the genes in the PCA to figure out which genes are more strongly discriminative in separating along any particular axis. Is there any questions about that? It's a quite useful technique. It's applied in a lot of areas. Of course, you need to have properly normalized data underneath this, which is, you know, a long conversation, but yeah, this is the essence of it. So you can see clustering of samples, which you can do some interpretation around. And actually, if you take the output of the algorithm itself, you can actually get the weights of the genes in each of these dimensions. And maybe there's some small set of genes that are strongly involved in pushing, you know, genes left and right, for example. And that would then allow you to make some interpretation about which genes are involved in distinguishing glutamatergic from GABAergic, for example. I don't see any questions, which is either a good or a bad sign. Okay, so
question.
Yes.
It says 37% variance and
Yes.
then 0, then plus 50 and minus 50. So for what does it stand for?
Great question. So this this variance is independent, actually. Well, it's not independent, but it's not the same as the scale here. So it's a good question. The scale here is something like information content. Yeah. I put the same dimensions on these so you, well, close to, so you can sort of see the dimensions in a comparable way. But what this is telling you, for example, if, you know, on this axis, let's say it only went from 0 to 5 on either side, right? Let's say it was about 10-fold less in scale. Generally, that would mean that you'd have less variance explained by this particular principal component. But the way you can think about it is that, you know, I was describing that each of the genes has a weight in terms of how it's collapsed into this one particular supervariable and this principal component. You can consider that as, you know, information content, yeah? And that helps define the magnitude of projection along this axis. So it's not a meaningless scale. It actually has a relationship to the amount of information captured by this particular axis. But it's difficult to explain beyond algebra effectively. So it has to do with the way that the genes are weighted. And there's a relationship between that and how much variance is explained overall from the data set. So you would find, let's say, if you're like 10th principal component, you have a very small amount of information or small amount of variance that's you can capture in that particular dimension. And correspondingly, the scale of this would shrink when you have less variance explained. Does that help?
I still have the question. So if the question would be, what tells you the blue dots? What would be the answer?
Could you repeat that question, please?
If there would be a question for what does the blue dots stand for? I mean, what's the information from the plot? What would be the answer?
So each of these points is a sample, right? So when you get your data back from a particular RNA-seq experiment, what you have is a matrix of, let's say, 20,000 genes, and then for each sample, you have a measurement for each of these, right? So you have some, let's say, in this case, 4, 8, 12, 16, 20, like 30 samples, right? So you'd have a matrix that's, let's say, 30 by 20,000. And what you're interested in is now trying to separate these samples out based on the principal components. And what this is saying is that, you know, these are all replicates from the same sample class, right? These all come from these neocortical VIP cells. And what this is demonstrating is that the replicates, so these were tissue samples taken from 4 separate mice, but from the same brain region, effectively. And they all cluster together. And they cluster separately from this set of replicates of these different types of cells from the neocortex.
Maybe to add to that in a simplistic way, you could say that it also allows you to say that, for example, this one sample here, I hope you can see the circle, seems to be a little bit gone astray from the other samples, right? But that the variance within this group as compared to the other groups, the other cell types that were analyzed here, like this one, is still relatively small, so that this kind of analysis can also help you flag samples where you would have to worry, should I exclude a sample from the analysis? So let's say you had a data point for this blue group that had ended up up here. I don't know if you see my little Easter egg there. That could be a flag to say, oh, maybe something really went wrong here, and I may have to exclude the sample from analysis. So this would be basically an unbiased way to spot that.
Yeah. You imagine if you had two pieces of the brain that are right next to each other, and maybe surgically you had some contamination from one or the other, maybe you would see that it's intermediate between these two adjacent, tissue samples or something like this. Now, just because there's a greater spread here doesn't mean it's necessarily technically a problem. It may just be that this particular type of cell has larger biological variance, like the transcriptome is a bit noisier in these cells, right?
Or just one thing, this is one of the experiments of the things that you're gonna do later. So if not everything's super clear, you can discuss with your tutor later. So maybe we should move on to the hierarchical clustering because we don't have a lot of time.
Yeah, I think we actually hit the time limit.
I'm going to suggest that we do the 3-minute version of hierarchical clustering.
Yeah.
Then we all have still some 20-25 minutes to recover, digest until we continue with the exercise.
Yeah. So the good news is that hierarchical clustering is not as abstract to consider, sort of geometrically, or algebraically, I should say. And the basic idea is that we have this agglomerative clustering of n objects. So what we do is we start with, let's say, sample 1. Let's make it simple. Let's say we start with a sample here, and then we compare it, against all of the other samples. And then we find that this sample is the closest, and so we define some distance between them and connect them in this tree. And then from here, we find the most closely related sample to these two, which ends up being this one, and then we draw another branch to it. And then we progressively build up this tree. Yeah. So this is what's meant by agglomerative. So you're agglomerating these nodes, and each one of these is a separate sample, let's say. And we build this tree-like representation of it. So to do this, you need a couple things. You need a distance between the objects themselves, which in this case is our samples. And then you need a way to measure distance between the clusters. And by cluster, I mean, for example, what's the relationship between all of these guys here to any other particular sample? Because as we collect these together, we make sort of another grouping. And so we need to define the distance between any two sample, and then a distance between the groups and other samples, for example. There's a bunch of different ways to do this mathematically. There's different types of distances and different types of linkage. So linkage is how we stick groups together. Don't worry about it too much. The short message is that for gene expression data, this Pearson distance, which is not identical to Pearson correlation, this works well for RNA-seq data for distances. And then we generally use this word algorithm for connecting the groups together. So then you end up getting a tree that looks like this. One thing to keep in mind with the trees is that you can rotate them around any of these nodes and they're equivalent, right? So let's take this example here of this orange set of 4 samples here. I can put let's say my mouse or my finger on this node here, and I can actually rotate them. So if I was to flip this, that effect would be that these 3 samples are now on the left, and this one sample would be on the right. So you can imagine this tree kind of spinning around here like a child's toy you'd have above your child's bed. And it's arbitrary at that point, because these horizontal distances in the tree don't actually mean anything. All that's actually calculated for the distance is the vertical ones, right? So if I'm interested in the distance between this sample here and this sample here, it's only the sum of this bar plus this bar plus this bar, right? So equally, you could think about flipping on this one, and I could actually put all the orange branches on the left and all of the blue branches on the right, and that would be the identical tree. Yeah, but it's a different way of calculating distances, and you can also get clustering. You should get a similar result to PCA in most cases, but it doesn't work exactly the same. So it's something that you can compare for clustering. You can also map a so-called heat map on top of this tree that actually shows you the sort of a color scale representation of what the data looks like to build the tree. And you'll see that this afternoon. I think I missed the graph that shows this, but it doesn't really matter. You'll have lots of time to explore it this afternoon. Was there any questions about hierarchical clustering? I think you'll have a chance to look at it in detail later. There's just one more sort of 1 minute point that I want to make to close this up. In terms of projecting the data onto smaller dimensional space, which is quite useful because, in an RNA-seq experiment, you have, let's say, 20,000 genes that you detect, and any particular gene is going to have a relatively small effect on a particular biological outcome. Yeah, so the weights are generally small, and so trying to make sense of each of these individual weights is not so easy. And what we're often interested in is what's actually driving the observed transcriptome that we've measured, right? And what drives transcription are transcription factors. That since we're with the Scheifele group, we should also say that the splicing machinery is, and the Dutch group also, obviously there's splicing things that are going on, but there's sort of upstream regulators that control, the gene expression values that we measure here. So one way to think about this is to actually look at the activity of the transcription factors themselves. So if we know that a particular transcription factor regulates, X number of genes, we could actually look at the activity of all of those genes together and then look at the level of the transcription factor rather than the individual genes. So this idea was packaged into something called ISMARA that was generated by Eric van Limwegen's group, also at the Biozentrum. And in your spare time, you might want to have a look at this free web tool for doing this, because I think it's a really elegant way of looking at RNA-seq data that allows you to explore the transcription factor space rather than trying to make sense of the individual genes themselves. That's the last point that I wanted to make, and there's a bit of time now. I'm happy to take any questions. And I should say as well that I'm easy to find by email from Scicor, so if you ever have any questions about anything related to this, feel free just to drop me a line sometime.
Yeah, thank you, Jeff. Thanks everyone for the attention. We rushed a bit through the clustering, but I think, as Jeff was already indicating, I think it will become very clear what that does for you when you run the exercise. I think it will be really helpful. And I think in the interest of time, unless they're really pressing questions now, I would say we break here and then continue with the exercise at 11. Is that okay for everyone? I assume silence is yes in this case.